It wouldn't be a success without you guys constantly tuning in every month, and so I greatly appreciate that. So I'm just so excited to have Colin here today, and I'm going to introduce Colin, and then the next voice you'll be hearing will be his. Per usual, I will be dropping the link down for your evaluation. Um, if you guys are interested in receiving any type of personal development hours for this webinar. So I'll drop that in the chat probably around 11:45 or so. And if you have any questions, you can drop that in the chat as well. Colin, I'm sorry, we'll be looking at that. And of course, we'll have our question and answer session at the end. Colin attended undergrad at University of Wyoming. He earned a Bachelor of Science in Microbiology and a Bachelor of Science in Molecular Biology. He earned a Master of Science in Biology from Eastern Carolina University in 2023. He was mentored by Dr. Ariane Peralta. His PhD research in Dr. Peralta's lab focused on microbial communities in soil and sediment that transform carbon and nitrogen. Thank you so much, Colin, for taking the time out of your busy schedule to assist us in our webinars. And Colin, you have the floor.
Right. Thank you, Michelle. Thanks for that introduction. Thanks for hosting me, and thanks to everyone for joining. Go ahead and share my screen here. Okay, I think that should be good. So I'm going to be talking about microbial nitrogen transformations in regenerative stormwater conveyance systems, or RSC. I want to start by thanking my funding sources, especially the NC Water Resources Research Institute that funded a year-long fellowship for me to do this work. And then we supplemented that funding with the ECU Water Resources Center and also an NSF National Research Traineeship. Just a disclaimer for that NSF funding, these are not the views of the NSF necessarily. All right, so here's the outline for today. I want to start with just an overview of regenerative stormwater conveyance for nitrogen treatment and stream restoration. And then I want to spend a lot of time on the nitrogen cycle and try to give you a more holistic understanding for the nitrogen transformations in RSC that are most relevant for that infrastructure. And then I'm going to talk about the Town Creek Culvert Project, which happened here in Greenville, North Carolina, and involved the installation of 2 regenerative stormwater conveyances with some different design features. And I'm going to present the research that we've been doing on nitrogen transformations in those RSCs. All right, so to start off, we know that microorganisms transform nitrogen in stormwater control measures. There's a variety of important transformations that they do. 2 of the important ones that have been talked about a lot recently are nitrogen fixation and denitrification. And these processes work in opposite directions, if you will. So nitrogen fixation is importing nitrogen into the stormwater system, whereas denitrification is exporting nitrogen or removing nitrogen from the system. And we also know that domestic and industrial activities can increase these nitrogen inputs. And so our goal with stormwater management at times is to decrease the nitrogen exports that are sent downstream to sensitive water bodies that can have issues like algal blooms and fish kills. And so the goal is ultimately to decrease those nitrogen exports. And so it's tempting to think of these as just chemical reactions happening in sort of an industrial system. But I really want to emphasize that these are biological processes that are carried out by microscopic organisms naturally inhabiting these different stormwater control measures. And so in conjunction with the nutrient input dynamics that we have, there's also urbanization and what's sometimes referred to as urban stream syndrome. So this has to do with the hardening of infrastructure, so the expansion of impervious surfaces like concrete and asphalt, which cover up those wetland environments that have high rates of nitrogen removal. But that impervious surface, those also speed up flows. So those create those flashy flows. And those flashy flows degrade stream morphology. They can cause erosion and channel incision and those sorts of things. But those flashy flows actually decrease the time spent in those wetland-like environments that would be removing nitrogen. So it's sort of this dual problem where we're decreasing the time and space for nitrogen removal, and we're at the same time increasing nitrogen inputs from different domestic and industrial activities. And regenerative stormwater conveyance is a recently emerging stormwater control measure that has potential to address both of these issues, the nitrogen inputs and the hydrologic symptoms of urbanization. RSC, it's a media-based stormwater control measure. It's a series of pools and riffle-weir grade controls. And underlying the riffles are these beds of treatment media, typically a mixture of sand and wood chips. These features do have a demonstrated hydrologic performance. So there's published research showing improved infiltration, increased hydraulic residence time, and increased evapotranspiration, and actually resembling even pre-development hydrologic dynamics. So one good example from the North Carolina coastal plain, published by CISEC and others. And really the goal of this nutrient treatment media is to have a prime environment for denitrification or nitrogen removal. So the idea is it's providing organic carbon in a low oxygen environment that water can slowly seep through. And so in theory, that would be a prime environment for this microbial process that converts that dissolved nitrate into these nitrogen gases that can leave the system, thereby decreasing nitrogen loads. But there's been a really variable range of nitrogen treatment performance reported for regenerative stormwater conveyances. So just in this one study, we could see a range of 16 to 37% decrease. There's other less peer-reviewed studies that have as high as a 50% nitrogen decrease. And then there's even a published study showing an increase in nitrogen export during a storm event. So in general, there's just a really wide range of reported nitrogen treatment. And so we want to better understand why that treatment performance varies so widely. Is it because of the environmental controls on denitrification? So namely oxygen, carbon, nitrate concentrations, temperature, hydraulic residence time. And then at the same time, we asked this question, could these pools in an in-stream regenerative stormwater conveyance system, could those support that nitrogen fixation process that would be importing nitrogen? In stormwater ponds, not regenerative stormwater conveyance, but in stormwater ponds, there is published evidence of nitrogen fixation. There is no published study yet that looks at nitrogen fixation in RSC pools. We do have a data set I'm going to show you later on. That's a single time point where we do look at nitrogen fixation. We didn't find evidence of net nitrogen fixation, but we do have a call for further research on that. We want to study that more to really be certain that we're not seeing nitrogen fixation in these in-stream RSCs. And so I want to talk about the nitrogen cycle and really highlight three processes that are relevant to RSC. So the first one is that nitrogen fixation going from N2 to ammonia, then nitrification going from ammonia to nitrate, and finally denitrification where we go from nitrate to nitrous oxide and back to nitrogen or N2. So this is kind of a simplified nitrogen cycle, but you can see it does bring us around full circle from N2 to N2. And these processes are commonly found in stormwater infrastructure. And it's this denitrification that's responsible for complete nitrogen removal and is really thought to drive nitrogen treatment in regenerative stormwater conveyance and other wetland-like environments. All right, so here's a view of that simplified nitrogen cycle. Let me get the laser pointer. Okay, so I want to start up here in the atmosphere. We say that the atmosphere is the major reservoir or holding area for nitrogen in this N2 form. So 78% of the atmosphere is made up of this N2, and that's a highly stable form for nitrogen. It's generally non-reactive, but it's these reactive forms of nitrogen that are required for all life. And so there need to be mechanisms to transform this generally non-reactive N2 into these more reactive forms to support life. So one of the oldest processes to do this, a biological process, it's called biological nitrogen fixation, and that's carried out by microorganisms. This is an energetically demanding process, and it's highly sensitive to oxygen. So that oxygen can actually damage the machinery that those microorganisms are using to convert that nitrogen into ammonium. So it has to happen in low oxygen environments. That can be soil or that can be water. There are examples from stagnant stormwater ponds, like we've talked about, where they form a low oxygen, low nitrogen layer. And so since this is a energetically demanding process, those organisms do rely on a carbon source, either external, or they may be making their own carbon. In the case of blue-green algae or cyanobacteria, they may be photosynthesizing as well as fixing nitrogen. And then there's also human sources of fixed nitrogen. So we're showing here sources of fixed nitrogen, human and biological, in teragrams of nitrogen per year. So we can see that biological nitrogen fixation on land and in the ocean is a major driver. Lightning fixes a small amount of nitrogen, so a non-human process, but relatively small compared to these other ones. Now combustion, this could be fossil fuel combustion, that could be biomass combustion, burning things like wood. That can have a considerable flux of fixed nitrogen. But then really getting into the big human sources, it's fertilizer production, something called the Haber-Bosch process. And so this is where humans figured out how to harness that atmospheric nitrogen, that N2, and get it into a reactive form, ammonia. And that's used to produce synthetic fertilizers. And another process here is agricultural biological nitrogen fixation. And what this is planting of legumes like soybeans or clover. And these plants, they are good at housing the microbes that do that nitrogen fixation. So it's still biological nitrogen fixation. It's just that we've scaled it up with these large operations where we're planting a lot of soybean, for example, and so that at scale can elevate nitrogen levels higher than they would have been previously. And once that fixed nitrogen is produced, it can be transported in a few different ways. And one of those ways is a short-range atmospheric transport. So these compounds like nitrate, nitrite, ammonium, nitrous oxide, they can be aerosolized and transported over short distances, then deposited through either wet or dry deposition. And so again, fossil fuel combustion can create some of those aerosols, biomass burning, and then soils and agriculture. And so a large source that's going to be relevant for us is livestock operations, where we'll have open livestock waste that's high in ammonia and some nitric oxide. Those aerosols can be transported over a short distance and then deposited. So that can distribute that nitrogen away from a point source of pollution, away from that single livestock feeding operation, and it can distribute it over a region. So then you can have non-point source pollution in the region surrounding some of those industrial activities. And Eastern North Carolina is a hotspot for this nitrogen deposition. So we're looking at total deposition here, both wet and dry. The hotter colors are more total nitrogen deposition. And you can see this coastal plain of Eastern North Carolina is a hotspot nationally. And there was a study in 2008 that really showed the major driver of that is these confined animal feeding operations where we have open livestock waste lagoons, and that ammonium and other compounds can easily aerosolize and be transported over this region of the coastal plain. That study showed that the Albemarle-Pamlico Sound is within the transport range, so that can be inputting nitrogen to that nutrient-sensitive water body that has some issues with algal blooms and fish kills and whatnot. And it's estimated that it was a source of about 100,000 tons of ammonium per year from that coastal plain region. And so Greenville, which I'm going to be talking about, is also in the footprint of this hotspot. So even though it's an urban environment, it could potentially be receiving some of these nitrogen inputs from nearby industrial and agricultural activity. Another way that nitrogen moves once it's been added to the environment is through runoff or through liquid. A lot of these reactive nitrogen forms like nitrate and ammonium are water soluble, or they can be particles that are moving along in the water. And that runoff can, of course, go to nutrient sensitive water bodies like the estuary. And it also accumulates in the groundwater and in the soil. And so you can have this legacy effect where that reactive nitrogen has accumulated and continues to be exported in the watershed, maybe even a decade after some of those initial inputs. So those inputs continue to move through the system. Now there are a number of ways. So once that reactive nitrogen has been added to the system, there's really this huge diversity of processes that transform that nitrogen. And so these processes can go in the forward direction, if you will, so moving this way on the diagram. But there's also processes that go in the reverse direction, if you will. And so these things can move back and forth. And so that's really where you get a lot of the complexity in the nitrogen cycle. So I'm not going to cover all of that, but I do want to focus on one of those transformation processes called nitrification. And so this is the transformation of ammonia or ammonium into nitrate by these nitrifying bacteria. This process requires oxygen, so it's an aerobic process. And so in water, that would have to occur in a well-mixed water column, typically closer to the surface where there's lots of oxygen. And this is also known to occur in soil and sediment, again, in some of those drier or more surface layers where there's plenty of oxygen. And now we'll get towards the end of the cycle where we're returning that reactive N to the atmosphere. So I want you to focus in this diagram on these denitrification fluxes of carbon. So this occurs in the ocean, and then there's another large flux of denitrification on land. And so this is where we're ultimately returning that reactive nitrogen to the atmosphere. Denitrification is really the main driver of this. There is one other process called anammox that is more relevant in wastewater. Like wastewater folks may have heard of that, but really it pales in comparison to this denitrification flux. So really this is going to be our main driver of returns to the atmosphere. And some of the main controls on denitrification are carbon, nitrate, and oxygen. So I want to start a little bit by talking about why does oxygen matter for this denitrification process. And so to do that, I want to start with something a little bit more familiar, which is why we use oxygen. So why humans breathe in oxygen. And so what we're doing here, we are harvesting energy from organic carbon compounds. And what I mean by organic is just these big solid carbon compounds. They have a chain of carbon. And so that's really what we normally think about in terms of our food, in terms of carbs, fats, and proteins. They have these organic carbon compounds. So we're eating carbon, inhaling oxygen, and using that oxygen to harvest energy from the carbon source. And then we ultimately exhale carbon dioxide. And oxygen, for this purpose, provides the biggest energetic payoff. And so in this diagram, the energetic payoff is going to be on this y-axis with higher payoff up here and then lower payoff down here. And so oxygen, the reason why all animals and humans are using oxygen for this purpose is it gives the biggest payoff. And so that's why animals are large. We can do expensive metabolic things, like move around and have like a large brain, for example. But there's a lot of environments on Earth where oxygen isn't around and microbes still thrive there. So how does that happen? Well, it turns out they can use different molecules besides oxygen for that same energy harvesting purpose. And so the next best option, if oxygen is not around, is nitrate. It's the next most efficient. And so now in this denitrification, I'm going to kind of use an analogy to breathing. So for humans, again, we're eating carbon, inhaling oxygen, and exhaling carbon dioxide. For these denitrifiers, they're kind of eating, again, organic carbon, inhaling, if you will, nitrate. That's going to be used to harvest that energy from the organic carbon. And then they're exhaling a few different gases, nitrous oxide, nitrogen, and carbon dioxide. And so this is respiration for those organisms. It's similar to our breathing process. One kind of caveat, this process could be fueled by something other than organic carbon. There's some examples where things like sulfide could fuel this process in more of a marsh, like marine ecosystem. But really when it comes to stormwater and especially regenerative stormwater conveyance, the carbon source drives this process, organic carbon, so wood chips, for example. Right, and denitrification, since its discovery, has always been characterized as a hot spot and a hot moment process, where we'll see areas of really high denitrification, and then you'll see areas and times of much lower denitrification. And part of the reason for that, these environmental determinants, the oxygen, nitrate, and carbon, are not evenly distributed in space and time. So those reactants will also have hot spots and hot moments. And then the organisms themselves, their lifestyle, they're able to switch between using oxygen like we do, but then when oxygen disappears, they can quickly switch to using nitrate. And that's sort of their strategy. So they're good at switching back and forth often. And so that can create some of that variability as well. And so this is part of the reason we think that observed denitrification rates in regenerative stormwater conveyance vary so widely, is that we may just be sampling different hot spots and hot moments. And so what we're looking at here, this is an example from a floodplain of a river. They're showing the denitrification activity here with the heat map. And so this just shows there's these hot spots that are kind of shifting around from sampling point, and you could have a hot spot and then very low activity right next to each other. And in this study, they also do this same map for like nitrate and carbon. And you can kind of see that there's hot spots of those reactants as well, and they're not necessarily identical to these denitrification hotspots. So there's just this big variation in space and time for denitrification. All right, so can we engineer ecosystems to get more denitrification so that we can balance out the inputs of reactive nitrogen from organisms and from industry and domestic activities? And the idea here is that we could place these high denitrification environments into targeted areas where there's high nitrate. And then we could try to balance out this cycle by increasing this denitrification side. And so we're going to try to look at an example of that here in the Town Creek Culvert Project here in Greenville, North Carolina. This was a large-scale project to change a lot of gray infrastructure over to green infrastructure. I'm going to focus on the daylighting and regenerative stormwater conveyance aspects of this project, but there really was a large watershed restoration associated with this project. This Town Creek watershed in Greenville, North Carolina is an urban area, about 66% impervious area shown in orange. And there's a large underground stream network here shown in red. And this reach of the stream that was restored with regenerative stormwater conveyance shown in blue here, this leads up to the Tar River here. So where Town Creek joins with the Tar River. And this particular stretch that was a buried stream was undersized for a lot of storm events. And so there was nuisance flooding in this area. And so that really prompted the daylighting and installation of this green stormwater infrastructure around 2018 to 2020. And so the daylighting took this reach of buried stream confined in this concrete pipe. There was daylighting, and now in its place, we have an in-stream regenerative stormwater conveyance. And then also downstream of that in-stream feature, there was another regenerative stormwater conveyance installed. So this one is at a stormwater outfall coming off this parking lot. And so this flows into Town Creek downstream of the in-stream RSC. And so I'll call this one the dry RSC. This is a smaller feature than that in-stream feature. And I'm going to mark the in-stream feature in this blue color and the dry in this more orange color. That in-stream feature treats base flow and storm flow, whereas this dry RSC is only going to be receiving storm flow, and then it will be dry in between storm events. Our research questions, we wanted to look at how seasons and storms influence nitrogen cycling in these RSCs and compare the dry and in-stream RSCs. Our sampling, we look at upstream and downstream sampling points to look at changes in nitrogen from when the water is entering the RSC versus when it's exiting. And we do similar sampling in the dry RSC, upstream and downstream. We also wanted to look at fully submerged sediment from the bottoms of these in-stream pools versus this sediment island that accumulated over the life of this RSC. So that's shown here. There was sediment flushing in and this island sort of developed. And what this also allowed us to look at is what we might call a stream bank environment that's fluctuating in water and oxygen levels versus a stream bed environment that's always submerged. We also wanted to ask how storm flow impacted nitrogen treatment compared to base flow. Based on some work from Mike O'Driscoll and colleagues right after the RSCs were installed, they saw that base flow is occurring about 98% of the year, but storm flow, where we're overtopping the riffles, is occurring about 2% of the year. And in theory, this storm flow is just flushing through the system without time for that nutrient treatment. So this would in theory be minimal nutrient treatment conditions. I'm going to start by showing results from a pre-RSC versus post-RSC denitrification potential. And so before the RSCs were installed, a team from ECU did sampling along Town Creek, and they sampled sites that are immediately after what was that buried stream, that concrete pipe. And so post-restoration, it's still this reach that is daylighted. So there was sampling then, and then post-RSC, we've been sampling nearby. So within the RSC now, and in this dry RSC that's right nearby some of those pre-restoration sites. And the method we used to look at denitrification, it's called the denitrification enzyme assay. What we do is we sample fresh sediment, and then in a bottle in the lab, we provide an ideal environment for denitrification. So we provide nitrate, we remove oxygen, and then we observe the rate at which that nitrate is converted into these nitrogen gases. So that tells us the maximum potential for that sediment. What is the maximum potential for it to perform that denitrification process? And so here's the pre-RSC versus post-RSC comparison. On the y-axis here, this is that denitrification potential. So that's the rate of nitrous oxide produced by that sediment sample. Up here is just the total pre-RSC versus post-RSC comparison. And we did see significantly greater denitrification potential post-RSC. Now, this post-RSC data set does include seasonal variation. So we sampled spring, winter, summer, and fall. And so those values are all included, versus the pre-RSC sampling is from a single spring time point. So I just want to point that out. In this bottom panel, we're breaking down those denitrification potentials into some of these sub-environments. So pre-RSC, we looked at water column denitrification, and then stream bank versus stream bed. And then in the RSCs, we have the dry RSC, and then that stream bank or that sediment island, and then the stream bed of the RSC, that fully submerged sediment in the bottom of those pools. And so what we can see is those in-stream environments, the stream bank and the stream bed, had significantly higher denitrification than all the other environments. And interestingly, that dry RSC has a similar denitrification rate to the stream bed and the stream bank pre-RSC. All right, so here is a look at all four of those seasonal time points in the RSCs, where we're again looking at denitrification potential. So again, that in-stream RSC is going to be in blue, dry RSC in orange. We do show that sediment island. We show those samples in green here, and those are included as part of the in-stream RSC. Just looking from season to season, we see the highest rates of denitrification potential by far in the summer. And then within each season, we see greater denitrification potential in the in-stream RSC compared to the dry RSC. We did statistically look at whether that sediment island had different denitrification potential from the submerged sediment in the same pool, and we did not find a difference there. So that sediment island or that stream bank environment is having similar denitrification potential to the fully submerged sediment in that same area. We have another line of evidence that denitrification is occurring in the in-stream RSC. So this here is monthly water grab sampling, typically during base flow. And we looked at nitrate, nitrite concentrations and ammonium concentrations. I'm going to start with nitrate, nitrite. Upstream sampling is in these pink upward triangles. and then the downstream site is in these blue downward triangles. And we typically see a decline from upstream to downstream concentration of nitrate. We see especially large declines in these warmer summer months, and then smaller declines in the colder winter months. And this relationship remains if we standardize for concentration. So if we use chloride to standardize for concentration, we still see those declines in nitrite. So that's another line of evidence that denitrification is decreasing nitrate loads in the in-stream RSC. Now looking at ammonium, we don't see that regular decline from upstream to downstream. And in fact, we had this strange month where there was this large spike in ammonium downstream. We don't really have an explanation for that. But in general, these levels, while they don't decline from upstream to downstream, they are generally pretty low. And so our thinking on this is that a lot of that ammonium is being oxidized into nitrite. That would be that nitrification process. And so we think a lot of that ammonium remains at a low level because it can be readily oxidized. But any ammonium that does remain, it does not seem like the RSC will further treat that ammonium. And that makes sense given what we know about denitrification versus nitrogen fixation. I mean, really the RSC is going to be a prime environment for denitrification. It will have some environments for nitrification, but it's really not designed to convert that ammonium into nitrite. All right, so we've looked at denitrification. We've looked at this side of the equation where we're removing nitrate from the water column, decreasing nitrogen loads. So where the RSC is acting as a sink for nitrogen. But there's that other process, nitrogen fixation, that we haven't looked at yet. And so that's that process that could be importing nitrogen into the RSC. And so we need to use a method that allows us to look at both the nitrogen fixation and denitrification. So to do that, we did a single time point of sampling where we sampled sediment cores from the RSCs, and we also sampled water column from the RSCs, the in-stream RSC. For the sediment, we run those on a time course and take water samples over 0, 4, and 8 hours. We submit the water samples directly from the water column and from these sediment samples for membrane inlet mass spectrometry. This mass spec method, it allows us to look at really small changes in nitrogen. So it's standardized to argon, but basically that allows us to see these small changes where nitrogen could be fixed or where nitrogen could be produced by denitrification. So it allows us to look at the two processes at the same time. I'm showing the results here for those sediment samples where we looked at net denitrification versus nitrogen fixation. And so this is that denitrification rate. And so all of the sediment samples from the dry RSC and from the in-stream RSC, they all had positive rates of net denitrification. So none of those sediment samples were fixing nitrogen. They were all removing nitrogen. And we saw actually the greatest rate of denitrification in the dry RSC. And that's in contrast to that data I presented for the denitrification potential. And so this may be due to a hotspot of nitrogen at that time of sampling. And we do have some other data from the dry RSC where we see these hotspots of nitrate at certain sampling points. And so that's kind of our thinking, is that this may have happened to be elevated in nitrate and that activity for denitrification. But in general, this is a good sign. So that sediment is continuing to denitrify and not fix nitrogen. And now here's the water column samples. And so the way we look at this is the ratio of observed nitrogen to expected. So if this ratio is greater than 1, it means the microbes are adding nitrogen to the water column, so they're denitrifying. If it's less than 1, it indicates that the microbes are removing nitrogen from the system or fixing nitrogen. When we analyze all of our points together, we don't see a significant difference from one. So we don't see either net denitrification or nitrogen fixation in the water column, despite some of the variation you see here. And there's multiple explanations for this. It could be that denitrification and nitrogen fixation were balanced, but a likely explanation is that the RSC pools are in fact well mixed enough. So they have that continuous flow, there's mixing in the water column, and that's sort of remixing this sample to expected nitrogen concentrations. So this is a promising result. We're not seeing large nitrogen fixation, which would be concerning, but it's a single time point, and we really need to do more research on this. But so far, the indications are good that we're not getting unwanted nitrogen fixation in the in-stream RSC. All right, and now turning to storm flow, we have multiple storm sampling time points for this RSCs. I'm going to focus on one recent one from Tropical Storm Ophelia. This was September of 2023. This storm made landfall near Emerald Isle and hit Greenville pretty directly. There were about 4.5 inches of rain in Greenville, and we had significant flooding along Green Mill Run, which is sort of on the opposite side of downtown from Town Creek. And now looking at the water levels from that storm in both the in-stream and the dry RSC, we saw the initial rising limb here, and then water levels peak around midnight on the 23rd. For the dry RSC, we again see that rising limb, and levels kind of remained high throughout that peak of the storm. What's interesting with the dry RSC, this downstream sampling point, it really did not rise in water level. The infiltration was so effective with this dry RSC that we really didn't see spikes in water level downstream. And so that's a positive sign for the infiltration of that feature. All right, so now we're going to look at some of the nitrogen concentrations in stormwater samples. These concentrations are all standardized to chloride to account for dilution. So looking at ammonium and nitrite, we see that large first flush in the in-stream. And then for nitrite, we see this second flush, which isn't as present in ammonium. So this, again, could be an example where some of that ammonium is being oxidized and going into this nitrite pool. In the dry RSC, we again see that first flush of nitrogen into the system. We were able to do downstream versus upstream sampling for only 2 of those time points at the height of the storm where the downstream pool actually had water. And in the first of those time points, we did see a drop from downstream to upstream. And so that could indicate some physical treatment by the RSC. Perhaps some of those particles bound to nitrogen were settling out. In this second time point, levels of nitrogen were pretty low upstream and downstream. All right, now looking at total dissolved nitrogen, again, we see that first flush, and then this smaller second spike for the in-stream, and then similar results for the dry RSC. So these are showing us really effective infiltration for the dry RSC, perhaps some treatment during storm flow, but we'd have to look at that more, get some more time points during storm flow for the dry RSC. And for the in-stream RSC, this confirms other data we have where during storm flow, you're not seeing a decline from upstream to downstream. So effectively, that water's flushing through the system, and there's not treatment happening during storm flow. All right, so in summary, nitrogen drawdown via denitrification is happening in regenerative stormwater conveyance. We tested nitrogen fixation at a single time point, and we did not find evidence that there was large nitrogen fixation happening. But we do think that should be further investigated by others, other time points, other RSCs, to really confirm that that process is not adding nitrogen that's unwanted. We see the greatest nitrogen treatment in warmer months. And then during large storms, we see minimal in-stream nitrogen treatment, but the dry RSC did effectively infiltrate storm flow and even showed some indications of nitrogen declines during the storm. I want to say a big thank you to a lot of folks. Thank you to my mentors, Ariane Peralta and Mike O'Driscoll, other collaborators on the project from ECU. Thanks to a bunch of ECU students that worked on this project at different times. A big thanks again to funders, NCWRRI, and ECU Water Resources Center and NSF. And I'd be happy to take any questions. And I'll just point out, any code or data that I've presented is accessible on these links here. You can scan the QR code. But yeah, I'm happy to take any questions.
Thank you so much for that, Colin. And I'm looking in the chat. Right now, we don't have any questions, so that means that you did an excellent job. I dropped the PDH link in the chat as well, so you guys should have that. And so yeah, it doesn't look like we'll give it a couple more seconds. And if no one has any questions, oh, there's one by Francine. How do the microbes live in a dry system when there?
Yeah, good question. So in general, we think these environments are just really inhabited with a really diverse range of microorganisms. So there would be microbes there that thrive in those drier conditions, in those high oxygen conditions. But then there could also be microbes that go dormant. And so this is the thing about microbes. They're good at going dormant, basically going to sleep when resources aren't around. And then they can wake back up when the resources they need are around. So for those microbes that need to be in a wet or low oxygen environment, it could be that when the dry RSC is dry, they're just in a dormant state. And then maybe when the storm flow comes, they're able to wake up and able to do some of that denitrification. Or it could be in the specific case of denitrification, the same organisms could be switching back and forth between using oxygen versus using nitrate. So the same organism could be versatile in that scenario.
Okay, I think you had another one from Joe. Does soil temperature affect the RSC?
Yes, absolutely. So just looking seasonally, we do see the impact of temperature. I'm gonna actually show that again even. So that should be pretty close. So here's where we see the impact of temperature, just in terms of is it summer or winter? So we have evidence of that, and that fits with our understanding of microbes. So we know in general, pretty much all of these microbial processes, they'll happen at a higher rate at a higher temperature. They're usually more active in that way. And then in terms of tying that specifically to water temperature or soil temperature, that is something we'll do as we continue to analyze this data. So we do have water temperature sensors in place, and so we'll just tie that directly to those denitrification rates. But in general, it's a pretty tried and true relationship that when you see that increase in temperature, you'll see an increase in denitrification or really any other metabolic process. So I'm seeing,
Yeah, I decided not to read them.
okay, I'm gonna try to get the next one. Yeah, I think I see the next one. Could the fall nitrogen spike have been caused by lawn fertilization? Yes, I think that's potentially an explanation. Yeah, that is a heavily landscaped area near the ECU campus, and so yes, it's really possible that fertilizer drained into that downstream pool and created that spike. Yeah, I do agree with you. I think that's a likely explanation, but hard to be sure in that situation. I'm going to try to go to the next one. How can we practically apply this in the field? Um, yeah, that's a good question. I think RSC is being looked at by others, and my understanding is it's the process to incorporate this into the North Carolina Stormwater Manual. That process is underway. So there's a lot of data like this that's going into that. Um, in terms of application, like in terms of deploying more RSCs, I think we could absolutely encourage more of that, especially in these urban situations where you want to be treating hydrologic symptoms, and then you also have elevated nitrogen levels. I think this would be a safe tool to use for that. Um, and then in terms of refining RSC further, uh, I think that will have to be looked at more, but just considering design features like the stormwater outfall versus in-stream, they'll have different trade-offs. In terms, we could increase the length of an RSC, you could add more pools, and that would ideally give you more treatment. You're having more residence time in there. So I think a lot of the applications, it's going to come down to research on these systems in different environments. These systems are pretty widely deployed in Maryland, and they're starting to become more popular in North Carolina. So I think there's plenty of examples to work with, and the research is continuing to look at this. So I think, yeah, this will continue to move along. I hope that sort of addresses that question. I'm going to move on to the next one. Are there RSCs being used in areas other than the coastal plain? Would this work in the Piedmont and the mountains as well? Yeah, that's a good question. Um, Most of the literature I'm familiar with is coastal plain, um, and especially like the Chesapeake Bay watershed was a lot of the early applications of RSC, and then yeah, definitely literature in North Carolina coastal plain. I think there are examples in the Piedmont. I think in terms of the hydrologic performance, I think there's still examples of that working well in the Piedmont. I'm not as familiar with a nutrient treatment study in the Piedmont, but my guess would be that it would still work well. Even if you have those large slopes, I think one of the features of RSC is that grade control, where the pools can kind of stabilize that slope. So my guess would be that RSC would still work in those more mountainous areas. Question from Kim. Recommend design criteria manuals for RSC to date. Yeah, so that's a good question. I believe the state of Maryland does have RSC incorporated into their state design criteria. So I believe that should be available from Maryland. And then my understanding is that for North Carolina, the process is ongoing. I believe Bill Hunt's group from NC State is working on creating those design criteria, and that's sort of being reviewed by the state in an ongoing process. Um, in terms of other design manuals, I don't necessarily have another example to date, but yeah, I think a lot of that work is ongoing. Is there a map available of RSC projects in NC? Not that I know of, but that is a good idea. Yeah, that I think that would be worth putting together. But yeah, I don't have a map like that myself. All right, we are seeing, so there are some examples of Piedmont RSCs that folks are familiar with. Is there any work being done to compare RSCs for treatment of agricultural runoff versus urban? That's a good question. I don't know of a published study, but I do know a research team at ECU, Guy Iverson's team. They are working with a livestock operation that's installing an RSC to treat runoff from one of the lagoons. So that study is pretty far off. I think the RSC still has to be installed, but I think that team will eventually want to compare that RSC performance for runoff in the agricultural setting, and then we could compare that to the Greenville RSCs. So yeah, I think that's a really good idea for a future study, but I don't know of anything like that to date. Thanks for sharing those resources from Anne Arundel. That's great.
Okay, I think that is pretty much all the questions that we have. I think you pretty much covered all of them. So yeah. Again, thank you so much, Colin, for taking out your time, and we greatly appreciate it. And thank each and every one of you for attending this webinar. And like I said, I'll get these PDHs out to you, your certificates out to you as soon as possible, hopefully by the end of the week. And if you have any other questions, if you want to go back and use this for reference, we will have this uploaded onto our training site by Friday, Monday at the latest. And again, you guys have a wonderful rest of the day. And thank you again for your time. I really appreciate it. You guys have a wonderful day. Thanks so much, Colin.
Thanks, everyone.
All right.
Have a good one.
Bye-bye.